Very good. Well, thanks very much for the invitation to speak today. And I'm going to talk about some of our collaborative work on the structure and design of coronavirus spikes. Start off with a little bit of history. So as we know, the surface of the virion is decorated with spike proteins that give the fringe that led to the name coronaviruses, because it kind of looked like the solar corona. And the most important function is that they bind receptors and then subsequently fuse the virus and cell membranes. Right. So the spike protein is a class 1 viral fusion protein. It's the largest member of this family. Right. So here you can see several. Here's the coronavirus, S protein, HIV-1 envelope, influenza HA, F proteins from paramyxoviruses, pneumoviruses. These are all synthesized as a single chain inactive precursor. The S1 subunit contains the receptor binding properties, and this is generally a fusion suppressive cap. And the S2 subunit here contains the 4 elements needed to promote fusion, which is the fusion peptide, the transmembrane domain, and 2 heptad repeats, heptad repeat A and B, that end up coming together in the post-fusion state to form a 6-helix bundle. So these look a little different, but if you align them by their cleavage site, then you can start to see how these family members are more related. So the coronavirus spike is largest, perhaps because it has the largest RNA genome. The HIV envelope is the GP120 component, which is similar to S1, is substantially smaller. For flu, HA1, which binds sialic acid, that's even smaller. And then the paramyxovirus and pneumovirus F proteins have essentially just lost the fusion suppressive cap. And they all contain the S2 subunit. The fusion machinery is pretty similar. The F proteins are actually really quite similar in length to the coronavirus S2. So all belong to this large family. This is the model for receptor-mediated triggering and infusion that we first proposed in a paper in 2017, where you have the coronavirus spikes in their pre-fusion state. They exist with their RBDs, maybe all 3 down or in equilibrium with 3 down and 1 RBD flipping up. They combine a receptor, which then locks the RBD up. As each RBD flips into the up state, another receptor combined until it goes into this 3 RBD up conformation, which is unstable. S1 subunit sheds. S2 then rearranges into this pre-hairpin intermediate, shooting the fusion peptide into the host cell membrane. And then this rearranges and collapses back down into the post-fusion state, bringing the host membrane and viral membrane together to form the fusion pore and allow entry of the viral genome. So I'll first start off about the SARS-CoV-2 spike structure, and then finish with our latest work on structure-based design of new and improved spike proteins. And this first part of the work was primarily performed by Daniel Rapp, graduate student, and postdoc Nian-Chuang Wang. So the work dates back to, I guess, 2014, 15, when we started working on spike structures, trying to get the first structure of what a coronavirus spike and this pre-fusion conformation that exists on the surface of the virion. And in 2016, 2 papers were published. We worked collaboratively with Andrew Ward and Barney Graham to determine the structure of the spike from the human coronavirus HKU1. And David Wiesler's group, working with Felix Ray, determined the structure of the spike protein from murine hepatitis virus. And overall, they were pretty similar, and we learned a lot about the architecture and function of these spikes. So here's a movie of the 4-angstrom cryo-EM structure of HKU1. See the S1 subunit in blue, S2 subunit in pink. The other 2 protomers are in white and gray. And see how the S1, we knew about the N-terminal domain and the receptor binding domain. There are some new domains which we were not aware of. We can see that the N-terminal domains are around the periphery, and the trimerization is actually mediated by the receptor binding domain. And so for HKU1, we saw really only this one conformation where all the RBDs were in the down state and would be inaccessible to protein receptor binding. And so we followed these other 2 domains, SD1 and SD2. The structure also revealed what the pre-fusion machinery looked like. We had a long central helix, which is common among the class 1 viral fusion proteins. And this was really helpful for sort of combining all the known literature on the biochemistry and biology of these spike proteins and how it relates to function. And this is what we could see for triggering. So we know that the S1 subunit, as I mentioned, is this fusion suppressive cap that sits on top of the S2 subunit in the pre-fusion state. And then after the protease cleavage and the receptor binding, S1 eventually sheds and comes off, and that allows the S2 subunit to refold. And it's this really dramatic rearrangement. So the central helices are in blue. Those roughly stay in the same conformation into the pre-hairpin and post-fusion conformation. Then you have all these elements of the first heptad repeat in green, yellow, orange, red. All of that polymerizes into a really long alpha helix on top of the central helix. And that shoots the fusion peptide in the adjacent region in magenta towards the host cell membrane. And to try and generate spikes for vaccine and for future structural studies, we wanted to try and prevent this triggering from occurring. We wanted to try and stabilize these spikes. That way they would always exist in the pre-fusion conformation and wouldn't spontaneously trigger and go to the post-fusion state. And so Nian-Shuang Wang in my lab used the structure of HKU1 and started performing some structure-based design, introducing several mutations. And what we were doing here was we were using the structure of HKU1 to create variants of the MERS coronavirus spike. And we had a hard time making the MERS coronavirus spike. See here, if we made the ectodomain of the MERS spike using the wild type sequence, we could get a little bit of protein. It's actually quite heterogeneous, as I'll show. And so one of the things Nian-Shuang did was start substituting these residues in this region between the tip of the central helix and the first elements of the heptad repeat, and substituted these with proline. Right, so that's in this region here, which is relatively conserved between HKU1, MERS, SARS, and the beta coronaviruses. And so substituting one residue to proline, we got a huge boost in expression. And then some additional residues to proline, we saw similar increases in expression. And finally, we decided to change two of these residues simultaneously to proline. And we went from a relatively small amount of protein to like a 50-fold increase into the protein. So we called that MERS S2P for 2-proline. And here's a different way of looking at the results based on gel filtration. So the wild type ectodomain barely produced any. If we look at area under the curve in red, there's essentially none. Whereas with the 2-proline mutations, we get a huge boost in expression. And then Andrew Ward's lab looked at this by negative stain EM. The wild type spikes, after purifying and shipping them to Andrew, were this heterogeneous mixture of pre-fusion and post-fusion, which we think is suboptimal for vaccine design. We wanted to have a homogeneous population of pre-fusion spikes. But with those 2 prolines, not only do we get the boost in expression, we also got this homogeneous population of pre-fusion spikes. And it was nice that the 2 prolines worked well for MERS, but also for SARS. So SARS-1, SARS-CoV-1. We could make a little bit of the wild type spike, but again, it was this mixture of pre-fusion and post-fusion. But when we introduced the 2 prolines in the same region, then we got this large increase in expression and all pre-fusion spikes. So we really liked this. So this was a universal method of stabilizing beta coronavirus spikes, and it worked well for HKU1, which didn't even need it originally. It worked pretty well for some bat coronaviruses that hadn't emerged. And so we were sort of ready with all of this knowledge for when the emergence of SARS-CoV-2 occurred earlier this year. On January 10th, the novel coronavirus genome sequence was released. By January 20th, my postdoc Nian-Shuang had cloned the gene, the spike gene, into our plasmids for expression. He included the 2 prolines in the same place as we'd done for MERS and SARS. By January 30th, Daniel had already purified the spike and started cryoline data collection. And on February 10th, we had the spike structure determined, manuscript submitted. So just 1 month after the genome sequence was released, we already had the structure determined, manuscript submitted. And a lot of that was because of our prior work on coronaviruses and knowing how to stabilize it. So the construct we made was the ectodomain, stopping at residue 1208. We had the proline substituted at positions 986 and 987, which would be here and here. And we also mutated the furin cleavage site to try and keep it a bit more stable. And so this works. This was our first time we tried to express the protein for SARS-CoV-2 with the two prolines. It worked okay. We got about 0.5 mg per liter, which wasn't a lot, but it was really pure, well behaved on gel filtration, and it allowed us to start the cryo-EM data collection and structure determination. And Daniel was able to purify the protein, freeze grids, and start data collection all on the same day, all on January 30th. And that led to cryo-EM reconstructions of 3.2 and 3.5 angstroms. And so this gave us the first look at the SARS-CoV-2 spike. And overall, it's pretty similar to the SARS-CoV-1 spike, share about 75 to 80% sequence identity. For our data set, almost all the particles had one of the RBDs shown in green in the up conformation. So the other two RBDs in gray and white are down, inaccessible for receptor binding. And so ACE2 would bind right here in this region, and otherwise generally look quite similar to SARS-CoV-1. Right. We always are missing some of the flexible stalk and heptad repeat 2, so it actually extends quite a bit longer here, but this region's generally disordered in the cryo-EM structures because it's really quite flexible. We could see a lot of motion, so if we analyze the cryo-EM data set with this 3D variability analysis, we can see the receptor binding domain and the one-up conformation is kind of moving in this up and down. And so it's not just a static structure. This is a top-down view. So as the RBD is moving in and out, we see concerted motions from neighboring NTDs and RBDs rocking in. So these are really dynamic molecular machines that are breathing. There should be some small fraction of particles where 2 of the RBDs are up, and maybe even a very small subset where 3 of the RBDs are up. With being able to make the pre-fusion spikes, we could show that it bound the ACE2 receptor really well. So ACE2 binding to the SARS-CoV-1 receptor binding domain, we determined had a KD of around 300 nanomolar, whereas ACE2 binding to the SARS-CoV-2 spikes, we saw a KD of around 15 nanomolar, about 10-fold higher binding affinity ACE2 for SARS-CoV-2 than the first iteration of SARS. And Daniel was able to generate some nice negative stain EM showing a single molecule of ACE2 here bound to the SARS-CoV-2 spikes. And so that confirms some of the other data from researchers who had shown that ACE2 allows entry of SARS-CoV-2. We could compare with the closest known spike sequence, which is from the bat coronavirus RETG13. So we see some differences in blue between RETG13 and SARS-CoV-2. A lot of the differences in the receptor binding domain. And it's now been shown by others that the RBD is actually most similar to a pangolin coronavirus spike, suggesting possibly a recombination between a bat coronavirus spike and a pangolin coronavirus. So really interesting. All the mutations are in the ACE2 binding site. The S2P version of the SARS-CoV-2 spike is now in 4 of the 5 leading coronavirus vaccines, or SARS-CoV-2 vaccines. So Moderna's mRNA 1273, that contains the proline substitutions at 986, 987. Novavax, which has the subunit based, their version, NVX-CoV-2373, also contains the 2P mutations at 986, 987. Johnson & Johnson has the adeno26. This is showing the 2 prolines and mutated furin site. So in their variant, which is called SPP rather than 2P, that contains it. And then Pfizer BioNTech, their BNT162b2, which is in phase 3 clinical trials, also contains the 2 prolines at 986 and 987. So it's been pretty exciting that a lot of the work we started with Barney and Andrew 5 years ago or so, and the design of these prolines by my postdoc are now in 4 of the 5 leading vaccine candidates. And so we're pretty excited and hope that this works. Some really beautiful data from Briggs Group using cryo-electron tomography on whole viruses showed that the spikes on the surface of wild-type variants look very similar to the spikes containing the 2P mutations. So overall, the 2P versions of the spikes look very similar. They respond antigenically the same to sera. And so introducing the 2 prolines hasn't caused any large conformational changes, and the spikes look very similar to what occurs on the surface of the virus. So we wanted to do a little bit better than the 2P version of the spike, because that was basically designed on a few hours in a weekend in January, and we didn't really have a chance to test a lot of the different mutations to see if we can make a better version of the SARS-CoV-2 spikes. And so in around March or so, as everything was shutting down at UT, we had some offers from help from Ilya Finkelstein and Jennifer Maynard, 2 professors here at UT, and they offered to have their students and postdocs help us do a second round of structure-based design. So that effort was led by my postdoc, Qinglin Shea, and grad student, Jory Goldsmith. And what we really wanted to do was try and make spikes that were even more stable and expressed better. So the S2P variant was okay. We got about 0.5 mg per liter, but it wasn't very stable in our hands. It didn't survive multiple freeze thaws, and we had a lot of feedback from the scientific community. We had shipped that plasmid to over 100 people. Barney had shipped it to over 100 people, and people just having a really hard time making spikes. So we wanted to do better. So Qinglin and I, for the most part, looked at the structure that we had determined back in February and came up with new substitutions to try and further increase the stability of the spikes. And so we focused on the S2 subunit, which is the region that undergoes a lot of the conformational change. We designed over 100 different variants. We focused on 4 different categories. One is the introduction of prolines to cap helices and stabilize flexible loops. We also made substitutions to introduce charged residues to form salt bridges, such as here. So this threonine to aspartate introduces a salt bridge with a neighboring arginine. Cavity filling mutations, where we mutate smaller residues like leucine to phenylalanine to help fill these small pockets that are generally found in class 1 viral fusion proteins. And then introduction of disulfide bonds to form covalent linkages between flexible regions, or a region that moves and a region that doesn't move between the pre and post conformational change. So collectively, the team, all 3 labs, students and postdocs cloned 100 different variants of the spike containing a single substitution or single disulfide bond. And all of them were expressed in 40 mil cultures. And if we didn't kill the expression, then they were purified and passed over size exclusion and also looked at by negative stain EM. And here's 25 or so that had some beneficial property. So this is, we're looking at expression in micrograms per mil. The variant, the 2 proline variant is shown here. And so a number of these actually boosted expression 3 to 4 times. And for class 1 fusion proteins, based on our work and others on HIV and RSV, Ebola, we can see that by stabilizing S2, you generally get an increase in expression. So it's not a bad idea to first screen for expression increases. Salt bridges did pretty well, so that we had a couple of nice salt bridges that expressed, that had increase in expression of 2-fold, 3-fold. Many of the prolines looked pretty good, some greatly increased expression. Disulfide bonds generally underperformed compared to what we had seen for other class 1 viral fusion proteins. Most of them killed expression. A couple gave us 2-fold increases. Cavity filling, we got some 2- and 3-fold increases. And we were pretty happy with that. The idea is that generally these are additive, and so even if we have several that only give 1.5-fold or 2-fold, if we start to combine all those, we can get really nice increases in expression. Right? And so this would be a typical gel showing our S2P variant expression here, and then some of the ones, disulfide that did boost, salt bridge, one of the really important proline residues greatly boost expression. So all of these were characterized by negative stain EM to make sure we were maintaining the pre-fusion state. Gel filtration, so the dashed line is the S2P variant. So these are all the disulfide bonds. So here's the one that boosted expression. The cavity filling and salt bridge variants, and then some of the prolines. So we had this nice collection of about 24 individual variants that either boosted expression or improved the stability. So then we had to come up with multi-substitution variants. And so we either tried to combine all the salt bridges, all the prolines, cavity filling and disulfide, or start mixing and matching to try and make the most stable spike possible. And one of the things we saw early on was that combining the prolines really provided this additive effect. And so we have the S2P variant expression shown here, a single proline, these combinations of 2 prolines, 3 prolines, and then combo 47 had 4 additional prolines on top of the 2 in our base S2P construct. You can see in gel filtration, the S2P variant is down here, and then as we go up to adding 4 prolines, we get a really nice increase in expression. The melting temperature shifts, so it becomes more stable, going from about 46 degrees to 51 degrees. And we really like this combo 47. So that's 2 prolines plus 4 additional ones. So it has 6 total. Call it Hexapro or 6P. And it's really a beautiful homogeneous population of pre-fusion spikes by negative staining M. It expressed very well at large scale. So we went from about 0.5 mg per liter to 11 mg per liter for Hexapro and 293 freestyle cells. In ex vivo cells, we are getting around 32 mg per liter. We know of one company via transient transfection with a new cell line that's at over 130 mg per liter. So the protein can really express. If we look at binding to ACE2, S2P binds with a KD of around 11 nanomolar, Hexapro binds with a KD of 13 nanomolar, so it's basically identical. It's much more stable than S2P, so not only does it express more than 10-fold higher, it's more resistant to freeze-thaw. So if we take our S2P construct, purify it, and then freeze-thaw, and do it 3 times, there's almost nothing left, and there's just aggregate and junk. Whereas we get about 80% of our Hexapro is left and all in the pre-fusion state. For room temperature incubation, S2P doesn't really like being incubated at room temperature for a day or for 2 days. Hexapro is totally fine after 2 days. We'd probably go longer. We could even heat it. So Hexapro could be heated to around 55 degrees for 30 minutes and still look fine. Whereas S2P after 50 degree started to fall apart and you get a lot of debris. We determined a high-res structure. So Daniel did this. We got about 3.2 angstrom cryo-EM structure of Hexapro shown in green. We compare it to SARS-CoV-2 in white. Basically identical in the S2 region. Didn't really affect any of these portions where we introduced the prolines. The 4 prolines we introduced, the density was really nice. They adopted the conformation we designed them to adopt. And most importantly, there's really no difference in reactivity with patient sera between the S2P version and Hexapro. So this is performed by Greg Ippolito and Jason Levinder. They have different patient COVID-19 convalescent sera, and they plated either Hexapro or S2P. And for each of the 4 different sera, the reactivity was the same. So we're really excited about that. Sort of skip that. So to summarize, our prior research on coronavirus spike structures allowed for the structure-based design of these 2 stabilizing proline mutations that work generally well for all the beta coronaviruses. These are now found in 4 of the 5 leading COVID-19 vaccines. Hexapro, which contains 4 additional prolines, is substantially improved, and it's been licensed to several groups, and we're working with one to help create a vaccine for low and middle income countries. In the future, we want to start working towards development of universal coronavirus vaccines. Main question of how to define universal. Can you get all corona, all beta coronavirus, or just within a lineage of SARS-like coronaviruses? And start identifying other epitopes besides those in the RBD and NTD, try and generate broadly reactive neutralizing antibodies. So this is really collaborative work. I want to thank Nian Shuang, Daniel, Qinglin, and Jory for leading the 2 efforts. Jennifer Maynard and Ilya Finkelstein and their groups for helping out with the large effort to design Hexapro, Greg and Jason for the sera studies, and then Barney and Kizmekia, who we worked with coronaviruses since 2013, funding, and Hexapro. So happy to take any questions.
Okay, thanks very much, Jason. I have a question here. Does your work suggest gain of function potential for SARS-1 and or 2?
No, I don't think so. All the proline variants essentially kill the function of the spike. So you can't make a pseudotype variant with the prolines because it can't fuse. It can't adopt the post-fusion conformation.
All right. Thanks for that clarification.
Yeah. We're not making super spikes. We're essentially inactivating them.
Yeah. So this next question then I think is pretty along those lines. This is fascinating. Do you think the introduction of prolines in the spike has an effect on virus biology?
Yeah, it kills the biology. We've done that. Barney's lab tested that in our 2017 PNAS paper. So they tried to make pseudovirus with the 2 proline spikes, and it just couldn't infect those cells. And I assume the 6 prolines is even worse.
Okay, so next question is, how does the affinity of Hexapro for ACE2, oh God, we've got too many questions coming in. I'm losing my place here. Hang on a sec. Yeah, how does the affinity of the Hexapro for ACE2 compare with the native spike?
Oh, it's identical. That's well, yeah, S2P binding to ACE2 and Hexapro binding to ACE2. It's 12 nanomolar, so which makes sense. The mutations are all down in the S2, and the receptor binding domain's way up here. So that's not going to have any effect on ACE2 binding.
Okay. So do the Hexapro versus S2P versions result in different antibody responses?
They do when used as an immunogen. Barney's lab just showed me that data, and Hexapro is substantially better, which was surprising. I actually thought it would be similar, but it's actually much better.
And can you say more about vaccine plans with Hexapro for the developing world?
I'm not sure what's public. We're working with a foundation who's trying to create a vaccine that can be grown in eggs and that incorporates Hexapro. And we're trying to support them for countries that have large stocks of eggs and can make influenza viruses. And so this would be a vaccine that can also be grown in eggs and produced quickly. So that's at least one of the development pathways moving forward.
And did you patent your 986, 987 double proline mutant?
Yeah, so that was patented back, well, okay. The patent application was submitted back in 2016. I don't think it's been awarded yet. We filed another patent earlier this year specifically for SARS-CoV-2. But yeah, that'll be up to lawyers and patent attorneys.